today I will talk actually further from yesterday. Yesterday Ajahn Savarak talk about introductions to the genome data, right? And it's actually how to get the omics data from the biological cell or the living organisms. And today, what I try to talk about is go from the okay, yeah, fine. Okay. So now we're gonna go from the omics data to more understanding and the innovations, and particularly in the agriculture. And what we try to do today is actually to answer the questions like yesterday that I think one of the students asked the questions like how we go beyond the sequence, right? So once we know the sequence, when we know the genome, what shall we get the benefit from those, right? And today I'm gonna try to show that, okay? And first of all, I would like to introduce a bit about the center where we are from. Actually this center, we try to utilize the data or the omics data and to gain innovations in the agriculture. We call ourselves the Center for Agricultural System Biology. And this center is located at the Bang Khut Thiang campus of the KNUPT at the suburb of the Bangkok. And this is what we are actually doing. Just a brief introductions. So we get the data from the omics data from the genome. Actually, that is where we brought from. And then with that, we try to do the omics data analysis. For what actually? For identify what is important genetic code that bring about the phenotype that I will talk about in detail a bit more. This is just to give you big pictures of what we are doing in the research. And then we not just only know the factors, but we try to learn how that factors control the behavior or control the phenotype. And that's why we call we doing the metabolic modeling, or we do the modelings of the biological and regulations inside the cell. And then we know that the phenotype or the behavior that the living organism respond to the environments is not just controlled by the genetic only, but it's controlled by the interactions of the genetic code and the interactions to the environment. It's just like us. One guy responds to the environment differently to the others. And this is how we do the modeling of the interactions between the living organism and the environment. And lastly, we know that the living organisms not stay alone and not stay lonely in the environment. They stay together like us, working together. Microbe is as well. So we do the modeling of the species interactions and how that behave and respond in the particular environment. And this is the whole scale that we have done in the center. And based on that, just would like to point out that from the single nucleotide, right, from the genome sequence, we can go through from the sequence analysis to the modeling in metabolism in the cell, and the modeling of the living organisms and environment interactions, and also the modeling of between the organism to organisms under the environment. And then we generate a lot of innovations according to the data.
Sorry to interrupt, but just a quick question. You mentioned multiple species interaction. So this is assuming, so we would be studying plant species that maybe in danger occur in the same ecosystem. So like we see that this plant and that plant go together.
Yeah. That is the idea. So basically, based on the concept, we try to understand the interactions of organism interactions under a particular environment. And now what we actually do in the lab really at this moment is the interactions between the microbe and the microbe.
So biologists do have like a distributional or probabilistic idea of, certain species being together? Did they have that kind of?
Yeah. Actually, there are a range of techniques to explore the interactions between them. Right now, what we have done is based on the mechanistic modeling. So it is a kind of a little deterministic process. But one another, they apply the stochastic and also the AI as well. But I mean, for each method, they have the pro and con for each one, and they answer the different solutions for that point. Okay. So for this slide, I can say I just try to point it out that with doing this kind of early fundamental thing, we can come up with the many innovations. Like for example, for the first one, working with the sequence, so we can get the target genes or the target factor, genetic factors that we should be modified, or we should be make the inference to get something, right? And then we can have like many the modeling support system, like a dilation. This is the name of the model that we do the model of which plants interact to the waters. So then we can have applications that allow us to predict how plants feel and how much water that we need to give for the plants and make the maximize. We have the model to predict the growth of the plants based on the leaf area. We have like the model to the method to show that what is the quality material for the plant growing. And lastly, we can have like the guideline or the model supporting system that can tell us like what shall we handle the interactions of the microbial interactions in the microbial filter to sustain performance. And this is like the example. Okay, so according to the topic that we say that we're gonna go for the omics to the innovations. And this is something that I would like to spread, especially for the early career student or the early scientist. So to generate the innovations for the data, the must that you need to have is the understanding, underline whatever you try to make innovation for. And when I talk about the understanding, what is the understanding? The understanding is how the genetics influence on what we are look like. We call phenotype. And also the response. Just like we have like similar genetic, right? And we have this phenotype, and let's say for example, I am a Dasawalat, also ladies, right? We have like a similar phenotype. Because we have like about similar genetic that is different from identity. But three of us may respond to the environment differently. It's like in this room, Ajasawalat may feel already cold, for me and not. For identity, he said, oh, okay, that's fine. Right? So this is something that we call visual. It's like how the living organisms responds to the assertions, stimuli or environment. And then the response in terms of that growth, that is of course, if they're happy, they grow well. If they're not happy, they grow less. And all this is called the thing that to conclude this is called biological regulations. I will come up with that for a second. And then in the second part, I will show you a bit on the cell modeling demonstrations. Like I think like if you have time, I can like make a hands-on say on the cell simulation. And lastly, I will show you like how we formulate such a kind of this understanding, right? To create the innovations on the cultures. So you can see that what I try to say here, I try to go on from the genome data, right? And then how the genome data allows us to understand the living organisms based on the understanding of the biological regulations, how they've been regulated and how they've been controlled, our phenotype and behaviors. And lastly, the innovation is all about the ability that we control the process or the system. Okay. For example, like how we control the growth of plants. How we say that, okay, I would like to grow the plants with the maximum growth, and this only giving this resource. Okay. Okay, let's start with the first one. Okay. For all of my research and what I'm gonna talk later on, this is all a simple questions. How the phenotype happens, okay? And okay. And this is like to show you like how the phenotype happens, right? It's what part of it is about the genetic, right? Do you remember this one? This is like what I can teaching you yesterday. This is a simple dogma. This is how the genotype or the genetic control our phenotype, right? Gene, right? Gene, when we observe all the genes, we call genome, right? When we observe all the RNA, which is the product of the gene, is the transcriptome, right? And when the RNA translated to the protein, we call proteome, right? That is the whole protein, okay? And this is called the central dogma. So we can think in such a way that, okay, this is the genetic material, right? This gene and the genome, right? You have this gene, so you're supposed to have this type of phenotype, right? But many times, your genes is silent. Sometimes you have the similar gene as in micro, but you go differently at macro. You action differently at macro because that genes in microorganisms, they express, they do the function select in the protein level. But for us, it's silent. It's not expressed. So it means that we cannot, for example, correlate with that if like the volcano microorganisms. This is for example. But you know, it's much more better if our life is much easier. The thing is, this is what Sorak said yesterday. I'm sorry, I would like to change my speak because I cannot see this one. Okay. Okay, so you can see this, like this is from the genes, right? From the genome. And this is the blue one is gene. That is what she said yesterday. And then it's transcribed to RNA, right? And this RNA go to be a protein. And when it be a protein, it functions in the metabolisms. It means they catalyze from the metabolite A to metabolite B and the enzyme. This is a simple sequence, right? And this is we call the cellular regulations. And this is actually obey the central dogma, right? You have been, when your gene is action, this is a function. And it's a, right, it's not that easy. You can see this. This is also another part of the protein that is very more about the transcription factor. And the transcription factor is very satisfying. That is a factor to control the transcription. What does it mean? It means that it's not like this direction of the regulation, but the protein that translates here go back to control the gene expression. This is the feedback from the protein action to the gene expression. So then, yeah, okay, we know it's good, right? Okay, it seems like, okay, we find out, we know that there is another, another feedback loop to control the gene expression. But the thing is, in each level of the regulation, like the transcription, how the gene was transcribed, how the mRNA was translated, the protein, and the protein work itself in the metabolism. It's not work alone. What does it mean? It means that to be for one gene expression, right? there are a lot of the components that control the gene expression. More than one gene work together, right? The gene that is packing in that will be expressed, and another thing that express protein called transcription factor, right? You have the transcription factor present to make the gene express, right? And if you remember yesterday, one guy asked about that. How about the non-coding genes? For that is non-coding genes, right? So it is in this level, right? And we knew that these are a controlled expression of the gene as well. And again, these are a random alone. In the level, in the layer, the non-coding RNA and non-coding RNA work together. And then it's controlled expressions of genes. And this is the kind of really the simplified pictures of the overall cellular regulation.
I understand that cellular, regulation is about, breeding pieces of genes to make the protein. But in terms of like the timing of when do we need each protein, is that determined in here or is that due to something outside of this?
In here.
In here.
Yeah. So actually, now I not come to that yet. Actually, but you were right. The regulation is going to be expression and temperate. So we talk about the time. That is I will put it that later. But now it's just this of him and a presence of the individual. And when we say regulation business, what kind of things or type that make all components present and make the process action. So we can think about if we thought the process is being expressed, so that it is expressed, that it actually make output, right? You have to have the transcript factors, you have to have the non-coding RNA, you have to have many components that to make it express, right? But what I ask is how the treatment at the same time. Okay, that is how. Okay, that is another factors. Okay, and this is like just try to recap everything about yesterday to make you have a proper attention and talk about the word afterwards. So this is like where I then talk yesterday. Then he talked about the gene structure, right? Once and week at 1955. That is the first guy who know the double-stranded and the double-helix structures of the DNA, right? Curiously, but we know Mendel, right? Okay, so we know the material active chromosome, right? And we said, okay, they have this material. Like big E, small E, and so the tall plant and the small plant. So that is one. But they don't know at all, like how the gene manufacturing actually at a molecular level, right? Okay, in our body, in our ligament, and how they transfer from one to another, right? And afterwards, that they know that okay, the chromosome is when you unwind it, right? So it's gonna be a really long, right? And there is something that is called gene on the DNA sequence, right? And this is like we call at the molecule level when we know the DNA structures. And then when after the genomic data, right? We know the DNA structure, we can unwind that, right? We can sequence it. Now we try to, access more, not at the chromosome level, but we know all the sequence and well in located in the DNA sequence. And then we, bring it back into the chromosome, right? And we now know Okay, this gene is in which chromosome? Okay, so that's why we can understand Down syndrome, the other genetic persistence. Okay, and then later on, come to today, once we start kick off with the genome protein, right? Now we can access the genome level. Now we can have many more high throughput technology. So if I go back to this one, you see, in the term of the regulation, which is the secret of the life of life, there are many more levels. Present the gene is not a whole answer, right? So then, not just only the gene that we need, but we need to know which or how many is what is those, how many RNA were translated, and how many translate properly were we going to work. So this is come to the era that we have like many throughput technologies like the genome technology, transcriptome technology, proteome technology, and the metadome technology, and so on. There are many more. Okay, and okay, let's begin. And this is for example. Yesterday, I got give you some example like of the couple of sequence, right? And this is one kind of virus genome that is, I think it's more genome. But you see the number of base is about 10,000 base pair. The big question is how we make the understanding of this sequence. How do we put what does it mean? Where the gene are, how many metabolic genes or how many proteins, how many enzymes, or how does this work? And in this organism, how it's look like, how it's go from this simple sequence. Okay. Yeah. It's important, right? But yeah, got it. What next? Yeah, you can describe it. It's good. Okay, let's discuss a bit. We will start yesterday. What kind of information that we contact? Yesterday you brought it, right? And what again? Okay. You know that this is like RNA, the enzyme of the let's and then what else? The function. Very good. This is function, right? For this sequence, we want to know the function, right? Now, let's say at least you know that okay, in this genome, okay, they should have this function, or they should be able to write it or have this genome, right? So we always express in the term of the availability of gene in the genome. In such a way that okay, this is the maximum capacity that the living organism could be, or we call the potential, right? For example, if you have the drought tolerability, so it means that okay, support this organism can tolerate to drought, but it may not, right? Because it's not expressed, so this means that it's not working. The gene is allowable, but it's not functioning. That is, we talk about genome. But if you have the transcription, the transcription data, you know that, oh, okay, this gene can be functioning, right? And if you have little more, so you can answer more like how it functions. Okay? Okay, good. And okay, and this is like the point for today that I try to connect. So it means that if you would like to access biological regulation, right? As you know already, there are the energy level, RNA level, protein level, metabolism level, right? And then you have ability to investigate or measure it, right? You have ability to measure how many genes in the DNA, right? Based on the genome technology. you have the capability to investigate the expressions of the gene. You have ability to investigate the expressions of protein or the presence of the protein function at a particular time. And also you have ability to observe how many metabolites that cell or living organism produce as a product of the metabolic protein at the time. Okay, so I would say, okay, you know the component now, you know many in, you know many in the expressions of the time, right? You know the number of protein, you know the number of the metabolites. The key question is how this work together, or even this is work together. That brings to the phenotype or the response of an organism. Okay, so I think this is linked to the question yesterday that we have the question like how do we know that this is and this is expressed as a certain? Okay, this is all about that. So the technique that we use to analyze all this, we call the system biology. So the way of thinking underlying the word system biology is that we're talking about the factors that determine the living organism's phenotype, the phenotype of the living organisms, right? The way that the system biological approach we think about this kind of the problem. They thought that for a particular phenotype, it's not because of the actions of one factor or one component, but rather with associations of this component that work together. What does it mean? It means that we talk about the plant that correlates to the drought. That is not because it's not the drought correlates. Now you understand, right? Why is it that is potential? It's potentially it would be able to correlate the drought, but it doesn't mean that it can, right? So it means that if they associate with another genes or another group and work together in the way in the rationale, so it's come up with the ability to follow. Okay, and that associations or the action that work together is we call the networking. So it's the name of the network is really depend on which you are talking about. If you're talking about the gene expressions network, so we call it the transcription regulatory network, TRN. If you're talking about protein, the association of one protein to another protein, we call it protein-protein interaction network. And if you're talking about the network of the metabolite conversion, we call it the metabolic network or the metabolic. Okay, so this is a kind of the pathway that describes your metabolism. This is the graphic. Okay, and yeah, of course, everything can be integrated and we can do the model. Okay, and it sounds like you know, in one of time previously, it sounds like it's mainly all we are talking about the wonderland that now, and I'm going to show you that even the research in our center, we can do a lot of this. Okay. Okay, and this is like the whole pictures that I'm trying to say. Okay, to decode or to encode the secret of life, you have to link between the genotype to the phenotype. But it's not only this, but it's the genotype that responds to the environment that brings to the phenotype that we call the physiology. Okay, and this is just like give you a big pictures of such a kind of this field. So we talk from the simple dogma, right? And now we come with like more complex system, and the technique or the I'll say the principle that you use to analyze such a kind of this, how we call it the normal biology. The normal mean it doesn't mean that it is really normal, it is really good, but it is part of the new argument. to think of the biology. Right? So we use in this class of the technology we have like bioinformatics. This is knowledge like you are studying yesterday. From the sequence to the function. It's you can be in the middle of class, like you have sequence, you know and knowledge the function. Okay. There are a range of the technique on the mathematics, but okay, in overall you can think about working from the sequence to the function. Okay. System biology from what I was talking about. From the sequence to the function, to the system function. Okay. So this means that we now try to use the way of integrating to understand how this phenotype can work together. Right? I mean the component in this phenotype can work together and make the system physiology. And now it's good to know that is what synthetic biology that is really what do now. Anything from understanding technology. So you can think about synthetic biology is one kind of approach to innovation because that is ability to control, not the plant control itself. Regulated in plant control itself, regulate something. But synthetic is something like that. We try to control the video of the okay, try to control the process. And this is just a trend of such a kind of the technique, right? You can see that like since the 1953, you know the structures of the DNA, right? We started to have the Human Genome Project, right? We can sequence we have many sequences, and this is come with the biomechanics, right? Because as I told you, like how to get the information from that sequence. And then now we have many sequences. You play with the classes today, there are big database and many organisms, right? So how we answer questions, how we can compose that kind of information to the understand the regulations, right? And we did the actions of this organization to respond to the environment. And then it brings the systemability, really a hot field in the 21st century. And then now I think like the way to go is, it's not friendly. Not just only home one only, but many home parents. And then ability to synthesis of sorry. The synthetic biology. Right now, we is in the era of understanding and to build the ability to control the system. Okay. Before I give you an example, any questions so far? Okay. If you have not get questions, so I will show you an example that we have done to understand single class species. Actually, in the center, we have done so many species, not just Class08a, but we have a long experience with it, and I think this is the complete picture that is good to learn for teaching. Okay, I will go along with this that we just talking about, right? Okay, now get to know Class08a first. Class08a ภาษาไทยขึ้นมา2 พลังทุกคนรู้จักเนาะ okay. And this is the really important clock in Thailand because we get the money from Class08a other than rice and mouse. Okay, we are number first in the world market to export and trade Class08a. So then the big questions for us is how to make or how to produce the we call the solid wood. more and more. Because if you have more solid root here, you have more money. Okay. So the interesting for the cassava is that you see the small root here. This is small. When they was young, the root is really small. It's like a lap ball, fine root. So when in general, in other type of plants, it's gonna go like that for long, until they die. But for cassava, it's not. They have ability to produce bigger root called the solid root or starchy root. So that it allows them to collect a lot of the carbon and starch inside this kind of the root, right? And the key thing is that how do we access the code of cassava of the solid root formation? Because not all roots come to solid root. Now they select to grow and or expand as a solid. If we can access that, so it means that we can get more money from higher root. Okay, and that is the question. Okay, so now we're gonna go from here in RNA protein, and then I show you how we can use this kind of information to access the secret of life. Okay, start from the beginning. Okay, yesterday we will have the sequence, right? And I think like one to get the sequence of the linear synthesis, right? So that is the metabolic protein, right? That one is actually the metabolic, right? So it means that when you get the sequence, you can, for example, reconstruct the pathway. So when you have a sequence, you can put that sequence into the pathway of the metabolisms to construct, like, if you're talking about the starch synthesis, the substrate of the starch synthesis is the sugar, right? You can change sugar to starch. You can construct the gene in the pathway. This is the kind of the gene that you put in the pathway. The importance of this one is you see this is actually for one reaction. It's one reaction. It's supposed to have one enzyme, right? To catalyze the reaction. And this is actually one enzyme. But this is the gene in cassava that been annotated to have this function. There are a lot, right? And then you see the color in the back. This is the color mean. We compare this gene, let's say gene of cassava, with the Arabidopsis, maize, rice, Japonica, various types of plants, to show that, okay, this gene presents in cassava and presents in others or not. Okay, and if you have a quick look on this, you can see that, for example, this one and this one. So it means that the protein for this enzyme and protein for this enzyme, they have a variety of the genetic material available in other types, right? But for this one, we call it very conserved. So it means that you can see by the color here, every plant have this kind of the genetic material. But for this one, it's just only the plants in the first column and cassava that are available. And this allows.
So certain things are common to all starch producing plants. You're trying to find something that's unique to cassava. Does that have to have a specific function, or just uniqueness is what you have?
Okay, that is really good point. Because I think this is the high school knowledge. You know that every plant can produce starch, right? They do the photosynthesis, right? And then can convert sugar to starch. Every plant can do it. So it's supposed that the pathway should be common, right? And it is. You can see that there is no genes in cassava that other than have. It's in common. But it's a way that they common. They common with certain type of group of the plants and another type of the group of plants. Okay, so it means that it's not 100% overlap. And now the question is, how you think about it? Do you think that cassava will have a spiritual functions rather than others? Based on the same pathway. But the thing is, based on the physiology, you know that not all plants have the sunshine, right? It's not all plants all. Because it's like now cassava have certain kind of this physiology that the other does not. When you look through this sequence, this pathway, it's all about the same. Yes, some of them may be special comparing to the plant in general, but it has no really starting in cassava. That is it. Okay, but in another way around, what we try to say is that maybe it's not because of available of a special genes, but the genes have a different sequence. Okay, what does it mean? Yesterday I learned a lot. If you need to find glass, right? You got 100% iron, but many times they be annotated to this function with smaller percent identity. Let's say we say that we set the same function at 80% identity. What does it mean? It means that another non-identical sequence could bring to non-identical function. So it means it works the same function more likely, but it works better. Okay, and this shown in this one. So, for example, you can see that for this one, this is like all the protein in cassava that show that it should have the same function in the metabolism. It's catalyzed one the same reaction. And when we blast, okay, but there are many types of blast, but it's a type of same thing. When we blast like all against the other organisms, you can see Arabidopsis, maize, rice, custard bean, potato, and turnip. With a zero enzyme, that comes from it. So we can class it. Like okay, this sequence similar to this one, this sequence is not that one. And this is what it shows is that with a single sequence, with a single reaction, there are the varieties of ability to catalyze substrate product. So, okay, for possible. If you ask by the genes that cassava have this ability to catalyze phosphate A to product B or not? Yes, of course, because you see this metabolism, you see the pattern, right? How about the others? The others can do as well, right? But the enzymatic protein that catalyze substrate A to the product of cassava and the others. Not the same exactly. And with that, this similarity is made an atom or the whole effectiveness different. And this is the biology. Okay, and this is we can access from the genome code. Okay, we use only the genome sequence to work on this research. Okay, and then if we have the constant from data, okay, now you're gonna about to know when let's say you just sugar for you to start. You see this one, this is the enzyme. Okay, the blue one, this is the enzymatic protein that catalyze this reaction, this reaction, this reaction. Okay, we can see this two is catalyze this reaction, this two catalyze this reaction, this two catalyze this reaction. Okay, and you see this one, the circle hormone. Is genes. And you know the linkage? This is how we can find this gene and this gene work together. Okay, and it work together to control what? Let's say for this one, to control the expressions of this gene. Okay, how many? 1, 2, 3, and many more. Okay, to control the expression of ATS1. Okay, what does it mean? It means that if we have this kind of the network, we call the network of the gene expression. Okay, when the gene expressed in the same time, right? So ATS1 working. Okay, and how about, let's say, for example, very simple, 1, 2, 3. If we say 1, 2, 3, okay? If one of the three not expressed, what happened? Yeah, ATS1 not expressed, right?
So each gene, once the gene is expressed, you get a protein, right? So the interaction network is multiple genes. So that means that the interaction is multiple proteins. So if you have just some protein, but not in the right combination, you don't get metabolome.
Exactly.
So the gene interaction, so let's say the gene expression translates to creation of protein. The interaction determines creation of metabolome. Can we say something like that?
Yeah. It's a kind of many to many in one layer, many to another layer, many to another layer, and many to many possible layers.
And in biological function, I guess I don't know if this makes sense to ask. Most of the biological function are performed not by protein but by metabolome. Should I say that?
Yes.
Okay.
That is another difference. So I just make the thing like much more simple. When I say that the solution some factors go back to influence in expression. But actually the non-coding RNA and the RNA can go back to control the expression. And again, metabolize can control the deep expression as well. Everything can go back. That's why I said
Very confusing. Very confusing, okay. Very confusing.
So I like to add more things in these. So based on the picture here, you can see that okay, it's so complex, right? So many in one in these windows. Even like you know that if like control the gene expressions of ABS1, you need like at least 1, 2, 3 people here, right? And you think that it's so complex, right? It's not one-to-one. If you don't have this map based on visual bioinformatics, what I see in order to get this one, you have to think that in the genome of the SAWA, if I would like to identify the pink one, as is the TLP, they have more than 2,000. The SAWA have like 30,000 genes in the genome about us, and 2,000 are functions as the TF genes. You see that? They have possibility of 2,000, but not all to control of this thing. Yeah. 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 Because based on normally and percentage they talk about practice level, right? But they didn't tell you that these enzymes working well. Yeah.
เป็นได้จริงว่า
They have the genes that affect function. That's in some members just open. Because it is that. Stop.จะสร้างเป็นที่สุดเป็นสิ่งที่ถูกอิจฉาด้วยเช่นที่พาร์ทชั่นเจอรี่ที่สโซเชียลที่สโซเชียลแต่สอนเป็นก็คือเราก็สอนสร้างเป็นเป็นที่เกิดขึ้นในวันนั้นเป็นความฝันใช่ไหมต้องเป็นเรื่อง需要多少记忆？เพราะว่าถ้าเราจะนั่งแบบเป็นคนที่เสร็จเราจะเรามีเพลงต่างๆเราจะในการเชื่อมที่เหมือนเช่นเราถือว่าโอเคเราเนี่ยมี3คนที่คอนโทรลใช่ไหมคะแล้วก็ได้คอนโทรลได้เลยที่ของเราเรียนเชื่อมแทนที่เราจะ modify ด้วยตัวนี้นะการ modify เช็คกับ theory เช็คกับแต่ไอ้ตัวนี้เนี่ยไหมมันโมคนเยอะมากเลย modify ตัวเดียวได้สังเกตไปเลยเนี่ยเลยใช่ไหมแทนที่จะแบบเรา modify ตรงนี้ได้แค่แต่เปลี่ยนชั้นนี้แต่ถ้าจะ modify ตรงนี้ไอ้คนมันจะเอาตัวมันจะเอาตัวว่าดีตัวถ้าเราเรียนเราต้องแบบดีโมดีคนที่อันนี้มันอยู่เนี่ยก็ตรงนี้ถ้าเป็นของนั้นก็แต่ว่าก็จะสังเกตไหมไม่เปิดแค่เป็นของงานมันมีสแต็กของความ
ซึ่งเป็นแบบไลบรารีว่าที่เราจะมีไลบรารีว่าทีนี้ทำอะไรแต่ว่าพอที่นี้อันเดียว
บอกว่ามันใช่เป็นตรงนี้เด็กแต่ว่าใช่เด็กคนก็ต้องสมัครเนี่ยตอนนี้คนยังไม่ได้ยินก็สมัครคุณที่ดาวก็ต้องเองก็รู้สึกดีเราไม่ได้ยินอันนี้ถ้าเราตอนนี้ก็ทำได้มันก็จะทำให้เด็กOkay. Okay. Now we have to level one. We have like theme, right? That you see like the diversity, like the potential of the theme to introduce the diversity of the living organisms. And now we have like the R&D that allow you to see like how, for example, for this productions from the sucrose to the starch could be controlled according to the gene expression. Okay, and then. Okay, the third level. So, when the RNA is expressed, when the gene is expressed, right? We thought that, okay, if I say that, okay, if this gene, let's say 1, 2, 3, if this gene, and this gene expressed, APS function happen. Would be fun. We thought in assumption that this three protein from the gene expression work together once it expresses. But again, one complication is here. Sometimes the protein need to work together, need to work with another compound, or need to work with the part to make them active. So it means that protein and protein need to work together, right? And this is show that when the expressions happen, it's not directly to have the function of that protein. Okay,
So they have to have the coincidence of the compound to make the reaction achieves. Okay, and this we call the protein interactions or interactome network. Okay, and come back from this one. Okay, this is sucrose to starch, right? There are many enzymatic proteins, right? The blue one. Now this blue one, oops, sorry, turn to be the green one. All the blue one turn to be the green one. See, and the orange one. This is the protein that needs to be interacted to make the green one. That's it. That one.
Sorry, could you say the whole thing again? Just repeat it, tell me when you don't know what time.
Okay, I say that when we, for example, for this one, APS1, right? APS1 is the enzymatic protein E, right? Okay, you can talk about this is one protein that catalyzes this reaction. And to make simple, I say that we have this protein function. We need 1, 2, 3. Sorry, 1, 2, 3 in expression, right? As a component. So then we say that at this level, if 1, 2, 3 is expressed, APS1 should make an action. We think it's a way that this gene can work spontaneously after they express. But actually, then some plastic proteins have to work together before they active and make an action. Okay. So then we have to have another map. How this protein, let's say the red one, right? Work together at the protein level. Okay, to make an action. To catalyze APS1 or to activate APS1. Okay, so this is the real research actually. That's why I say that this one is also the real research. Okay, we knew that APS1 all the starch gene, we call it enzymatic gene of the starch synthesis, all the blue one here, have control by these transcription factors. And now we show another layer that the blue one in the previous slide to the green one here. It's actually the same protein. From the blue is presented as green in this slide, and the orange one is the protein that need to work together to allow the protein, this protein action in the metabolism. เข้าใจไหมคือโปรตีนแต่ที่เราได้โปรตีนเนาะเราคิดว่าโปรตีนเนี้ยมันทำงานกันคืออะไรมันทำงานได้หนึ่งเอานะโปรตีนที่ควบคุมการแสดงออกของยีนสีฟ้าของโปรตีนสีฟ้าถ้าเขาเอ็กซ์เพรสออกมาแล้วเราคิดว่าเขาจะมา activate การทำงานของ APS1 ได้ทันทีแล้วเมื่อพบว่า1 2 3ตัวเนี้ยเกิดการทำงานเราคิดว่า APS ทำงาน APS1 ก็เราคิดว่ามันทำงานได้ทันทีแต่ในความใช่มันมีการคอนโทรลอยู่แล้วมีคอนโทรลที่ในโลกของเราติดใช่หมายความว่ามีโปรตีนที่ไม่ใช่ transcription factor แล้วก็ไม่ใช่โปรตีนที่เป็นเอนไซม์แต่ว่ามันจำเป็นต่อการที่ทำให้สีฟ้าเนี่ยที่เป็นเอนไซม์โปรตีนเนี่ยไม่ได้ค่ะประมาณนั้นซึ่งจริงๆก็มีโปรตีนหลายชนิดเลยเนาะอาจารย์พูดแบบรวมๆแล้วกันมันก็เลยทำเป็นน้ำตัวนี้ออกมาให้เห็นให้คนที่มันเห็นว่าเนี่ยคือสิ่งมีชีวิตที่มันคอมเพล็กซ์มากว่าตะเกียบไอ้สีฟ้าตะเกียบจริงๆพอมันมีเซนในกราฟนี้มันจะเป็นสีเขียว เนี่ยคือสารอีกเป็นเอนไซม์อีกเอนไซม์อีกเหมือนกันค่ะที่หรือโปรตีนคือเราเลยเราพูดสั้นคือหมายความว่าตัวเอนไซม์เป็นโปรตีนเนาะเราก็เรียกว่าเป็นเอนไซม์เป็นโปรตีนกินในหลายครั้งเราก็จะพูดฟอร์ช็อตว่าเป็นเอนไซม์กินไม่ออกนะคะเวลาแอคชั่นเป็นโปรตีนเอนไซม์เป็นโปรตีนค่ะแต่เอนไซม์เป็นโปรตีนกินคือกินที่เอนโคดให้เอนไซม์ค่ะ
Can I picture the whole thing like there's a big network, right, of genes that may be expressed together, right? But they don't always express together. It's like, you know, a whole group of friends in a network, but a particular party maybe have 10% from that network. And when that gets expressed, you get some function, you get some metabolite, you get some protein, you get something. And so maybe the yellow represent all possible connection. And then the green is a particular instance of being expressed together. Can we say that?
No, it's not that way. So for the interactome, we try to say like the hybrid that allow one protein to action. It's just like the grid expression. But for the function of the protein, mostly it is action at the metabolisms or it's actions at the, let's say, the signaling pathway or something. But it is like a kind of thing like, you know, to make the protein A function. So you need the protein 1, 2, 3 to work with the protein A. You
So
can,
you need a combination of proteins. It's not just some random presence
No.
of protein,
no,
but this protein, that protein has to work in a specific way. It's not like a mixed distribution. They actually have to sort of like hold hand and connect.
exactly.
So
So
kind of like hemoglobin thing, where you need protein to hold hand and something.
yeah, that is the sequence model of how it's work together. So again, come back to the modern website, you know, you have to have this kind of component. We know that we have to work together, right? But the way that it's work together is this is the counting. This is not counting. This is counting component. Okay, so it is a kind of full mechanism, right? Let's say in vivo. If you would like if the protein A one atoms, you have to another protein to work together and make some protein A. But the way that it be together is not exactly together at that time, actually, but it is a kind of okay, 1 and 2 interact with each other, and then it's bring to the third one, and the third one is a scaffold of the 4, 5, 6 or something like that. That is the whole mechanism.
So you still need some random mixing for them to come together.
Yeah. It is not fully random, but it is.
It is, yeah.
It is some kind of.
So the real name is the name of
Yeah.
protein of protein chains of
That is the name of the enzyme. So you can think about the name of proteins. Okay. And that is what we talk about in terms of that is that we try to learn how to work together, right? And okay, so what I'm talking about is from one of this level, and one of this level, and one of this level.
to like very simplify this one, like for my understanding,
Uh-huh.
am I right? Like so it's basically like the genes, the like for example, you say that the start gene and the Cassava works, like they may exist, but like in different variations, but like they're not complete. So like it basically like change them to make them worse, like just modify the gene so like they can start like basically the whole thing.
Exactly.
Okay.
And what I try to show you the complexity like all the way through, is I just want to show the regulation actually. This is the only one that I try to use. But in terms of let's say biological regulation, bio is mean in the region of animals. And in this complexity. So to link with the data that we solve omics, genome, transcriptome, proteome. That's why people say that okay, if you have like no omics data, you learn more, and this is what we try to learn. But in the such way that you learn, it's not just learn only by the genome sequence, learn only by the transcriptome data, learn only by the gene protein expression. But the thing is, everything works in the text. So we can link it at a jigsaw. Okay, and when everything, let's say, for instance, is complete and then the process. That's all. That is simplified. And how all the factors or the cells that make the thing come together and form it to be action. That is part of the way. Okay. And then you can see that it's not very easy, right? To get or understand the regulation. That's why I say understand. Because you have to, do the research, like thinking about it, like what does mean. And then it's not be able to just do the data analysis. We have to use the simulations or the model to help to integrate all such of this together. Okay, that allow you to understand, the cost level regulations of this and allow you to link from the genotype to the phenotype. Right? Why the genotype is like look similar, but the phenotype will not the same. Why the genotype similar, but phenotype look like the same, but the next one to the envelope, which is physiology. Okay. And this is like what we try to say. This is like we do the model. It's because it's beyond our the calculations and analysis in the analysis. And okay. When we try to make a model and do the integration, this is something that we get. It's the same notation that this is how a gene, you can see that the node, all the node for genes, right? This is based on gene expression data. This is how the gene works together, right? And the one that highlight is mean this is the gene that will action in a particular condition. Okay, so you can see that when the plants go in the dry seasons, that is very hot and dry. And the plants go in the wet seasons that have more available of the water. So you can see that even they have the same genome, right? But gene will express differently. And plant use a different set of genes to handle with the environment. Okay. And this is like something that we get from our data. And more than that, when we have a model, okay, let me link to you a bit. You have a sequence, right? And then like you can construct this kind of the pathway. So now you can know that how sucrose, which is the sugar, can synthesize to be starch in cassava vessel. Okay, we already passed that part, right? Now I'm going to show you that not all the reactions in our metabolism work at the same time. In one time, this reaction work. In this time, this group of reaction work. In this time, this group of reaction work. Okay, so the questions that I try to answer is like, how the sucrose, synthesized to be the biomass at the different part of the cells. Okay, and we show that at the 3-month-old, 6-month-old, 8-month-old, can see the different metabolisms. Okay, we use metabolic model, and this is like the pathway. Okay, you can see the big pathway what is same, right? Because they have same genome. This is Class08a, right? And at 3 months, you see the thicker reaction, thicker line here. That is like when the sucrose have been synthesized. That is the major part. Okay, so it means that at 3 months old, okay, to give you more information, Class08a is about a year old plants. So we harvest like after 12 months. Okay, so at 3 months old, it means that it's very young, right? Can have not yet have the solid in the macro stage. Okay, so the way that they synthesize the sucrose is from this direction. Okay, you can skip the detail for now. Just keep so that okay, this is like the way that it's called. Okay, and they have just only this biomass. And this is why the research when we do the model. We know that when store up to the 6 months, where the starchy group have been formulated a lot. We found that sucrose have been metabolized in a different way. Okay, and it's going to starch, right? But the biomass number of another slide inside of the sperm is not yet high, but the starch is a lot. Okay, and this is the 8th month. This is when about the plants were harvested by the farmer. Okay, you can see that it sheds to this root again, but it's not formulate sac, but it formulates the biomass.
Sorry, what's the long thickness represent again?
The rate,
The rate.
yeah, the mass rate of the carbon synthesis through that reaction.
So it's like schematic of the reaction. But at different ages, probabilistically or in terms of average, a lot of it happens in the thicker line.
Deterministically.
Deterministically.
Yeah, this is like simulate based on the growth of the plants and sucrose harvest slide and the carbon dioxide output slide at that age. Okay. And then with that simulations, when we change the variable from age to the cultivar, so we know we can compute this kind of stuff that okay, we can access y 5 นาที which is one cultivar of the Class08a have low yield and the P50 have high yield for difference in terms of production. Because they catalyze sugar in the different way. And this is like the information scale.
So in this one, you can see that like into months, the HN is carbon 5 นาที which is low yield. And KU50 is the high yield. So based on the genetic information, both of them are very clearly
About the same.
Yeah, but while this one is very low yield, and this one is higher, even we grow this and this at the same area, this is the answer that we found. Because based on the metabolism, you can see that like the low yield, they use different enzyme to utilize sucrose and to get the biomass of the starch. And you can see that the high yield one, they use like the efficiency and it can know that what enzyme that is active in order to make KU50 make more starch. It means that based on the genome, you know all possibilities. You know the pathway of possible enzyme. But based on simulation, you can know what is the active one. What is the actions of that enzyme? Okay, this is the way that we see.
Okay. Okay. So, that is about one organisms. That is about the energy, right? Not it, the environment. Okay? Yeah, now we have outside that organism. Okay? So, just simply, to make it, allow us to understand the, physiology, that is the response of, organism to the environment. Okay? We implement another model. Okay? And that allow us to replicate the growth of the solid blue. You can see that is the real one, and which is actually replicated. It's based on model condition. And then it allows us to simulate the growth of the solid root by the time. Okay. This is like have been simulated by the when the carbon go through by the carbon dioxide fixation, and then by the time we can simulate the solid development according to this one, and this is like how we validate the our model. Okay. This is like we can simulate every day, right, until like from 7 days to 84 days, right? And then we compare with this one and this one at a time, and it seems like the amount of the of mass and the shape. This is like we calculate based on the carbon economy, and this is like allow us to use this model to predict how the solid would actually formulate under the list one. Sorry, under the factors of the thing. Okay, and this is like the first trial. Just have a quick picture. So everyone know that has one light to create the glow. Really important in the light. So we try to make a simulations that explain in the normal light. This is the amount of the root being produced by the plant. And if we grow the plant in the model, with the low light of plant, and this is happen, you will have a smaller biomass of the root. Okay, and this is like allow us to predict the production yield according to the light condition, according to the water condition. Okay,
And with this, what is the back? So based on the simulations, what is the applications that you can use is the budget?
Optimize the writing.
Yeah.
Yeah, if you would like to go to something, because we do this simulation type of software when we try to do the experiment in the Greek course, and we found that we could not get the solid when we blow a solid greenhouse, because I mean the further hypothesize that a long time that because of they don't have light to convert carbon to the biomass, but no one prove it, cannot prove it because the inside process very selective. And a viral model that that is clear because light is independent. You have no light on now. The energy produced convert the carbon to the biomass. It's not enough.
In terms of technology, this is probably more precise, right? It's not just more light everywhere all the time, but it's
No.
when, at which age.
Yeah, exactly. Yeah, I actually skip this part. So this is actually the detail. This is actually the representation that okay, we can simulate this. This is like aligned with the what we observe in the greenhouse, so on. And this is what the detail. We already showed that the carbon have been used differently in the sugar development and wood development. You can see the partition.
Let me make a comment of that students. This is basically the theme of your kind of approach. I think what bonsai students are going to be good at is you're not going to be finding the basic science. Basic science will be done by professional scientists. But what it means is that their finding gives a map of how you can optimize it. Light costs money, light costs energy, light is energy, energy costs money, you have to use it. And so this is why things like precision farming, so all kind of stuff that's happening in nature or in industry. The more you know about the computer mechanism, the more precise control you can make. The more precise control you can make, that's where your innovation comes in. Because now, you don't turn on the light all the time. Okay, you can precisely economize on the energy used to, grow cassava in this case. But this is just, you don't have to care about cassava itself. There's just one plant, but this is the theme of your study. Whatever technology you're in, that's your sweet spot. The sweet spot is studying the basic side, and I would say studying it in the commercialization, but it's in the optimization of the science. And that's why you get the basic.
Actually, I know that in everything are really complicated, but based on Ajan Sola and production yesterday, the thing that I try to active, because I got more time for things that is suggested. I try to read with your course, like why the technology needs to be done. Okay, so that was my step to lead to from really basic to something. It's not like I'd say ขอ experiment สุดท้ายแล้วเนาะคือตอนเนี้ยหลายๆคนก็บอกว่าได้ยินก็มีคนแล้วก็คิดว่ามันดีแล้วก็บอกว่าอะไรอย่างเงี้ยแต่จริงๆแล้วด้วย power ของมันไปได้เยอะมากเลยแต่การที่คำถามก็คือว่า what is the real goal I just like have class or have go for one pathway I think one pathway I don't know if this is how it's good well how it's like a little but that is reason I make trying to show that okay start from very basics from the state and then okay I think I will give you like 5